That's okay. That's no
David's
problem.
also away from his home.
Yeah, no, it's not a problem. Okay, we're recording to the cloud. Great. But I'm sorry, Madhur, because I think if this were happening on a weekday, we would have a larger crowd, and we're missing a few key people. So thank you so very much for your flexibility. And I think we can get started, and we're delighted to welcome you. By the way, the number of people who normally show up is not vastly more than this number, Madhur. And arguably, the really important folks are here. Maybe a couple of exceptions. We're missing a couple of grad students who may have other obligations. And but thanks to you, and thanks to all of you who are here, making time on a weekend. Madhur, I'm going to introduce you. I should say to you, Madhur, that rereading your abstract, you should be aware that some of the people here are kind of beginning level students. And so let's take it easy. And for me too, like what does ergodic mean, and certain terms that are going to be unfamiliar, but you're a professor, you know. Okay. So it is my really great pleasure to welcome Professor Madhur Mangalam, who is assistant professor and director of the multi-scale modeling lab of the Division in Biomechanics Research Development in the Department of Biomechanics, and Center for Research on in Human Movement Variability, all at the University of Nebraska at Omaha. And the way I got to know Madhur a little bit anyway was something that I can envelop in a bit of advice for all of the aspiring professionals here. And Madhur, I'm not going to go on forever, but I just wanted to share that Madhur is tremendously busy and productive, but he's also, in addition, he's done this very special, unique thing, which is to create a podcast where he interviews scientists who work on relevant topics. And I think that's just a wonderful service. And it also affords tremendous networking opportunities, such as here he is, basically a starting out young professor, and I now know about him, and I am now introducing him and have invited him here. And so what goes around comes around in a very good way. And I think it's a very inspiring example, Madhur, what you've done. I had the pleasure of interacting with Madhur and introduced him, asked him some questions for a while, and Madhur just put up the podcast link. And I think the name of it is BeyondPhreology, with PH, phrenology is the study of the bumps on your head and how that relates to your talents or lack thereof. So Madhur is ready to go, I think. If you have some kind of visual you want to share with us.
Yeah, I'll share the screen.
Okay. And Madhur, the way we normally go is that our speakers present for about an hour. Then we take a short biology break for a couple of minutes, and then resume the discussion afterwards. So that's how we're going to do it. Okay. So thanks again, Madhur, for joining us.
So thank you, Professor Rosenbaum. It was a very kind introduction. And for all of you, I'm Madhur Mangalam, assistant professor of biomechanics. We have probably one of the only departments of biomechanics in the country. Mostly what we do is studied under neuroscience departments or under human movement sciences or kinesiology departments. So we are uniquely focused on biomechanics here. And we mostly deal with human movements. We do a lot of neuroscience kind of experiments as well as the biomechanics kind of experiments, which include rehabilitation, development prostheses, and making them more acceptable, for instance, for people to identifying, how we can improve these, assistive devices for different disabilities and things like that. So we have a pretty broad spectrum and collaborative work, within our department. Most of my primary work is in that direction, and I specialize in multi-scale modeling. So by multi-scale modeling, I mean here multi-time scale and multi-space scale. So when you're looking at, for instance, a behavior, our behaviors are not unique to a particular time movement. So for instance, I'm giving a talk. This talk has some kind of roots in the podcast which I did with Professor Rosenbaum before, right? So it had this prospective aspect to it, and at the same time, your past determines future, and your future is also tied to the past. Your future also influences the past, given that when you plan things, that planning itself incorporates some constraints in what you're doing currently. So there are specialized techniques of time series analysis where you can look for these interactions, and you can identify things which are not yet happened. For instance, a person is walking and the person is likely to have a fall, right? So how you can use the relationship between past and present to identify those unknown events while they have not happened yet. Now, I just received a question, what's the difference between multi-scale and multi-dimensional? So multi-dimensional, when generally speaking in a typical language, are when you're considering 3 or 4 different aspects of something, or you're talking about a space. So for instance, you can have a behavior in 3-dimensional space, an ant moves in a 2-dimensional space, that's a spatial 3 dimension. You can have it's a very interesting question. So normally when we talk about dimension, we talk about factors from a very Euclidean kind of thinking. So when you're talking about space, the x, y, z component, there are 3 perpendicular orthogonal factors, right, of space. We have a 4th factor called time. You can have a multi-dimensional data set, for instance, when you record from the brain, when you record from the body, and you measure 5 different things, and you can call them 5 different dimensions or something, right? And you can map any particular behavior along those dimensions. So for instance, we know like things like LLM and ChatGPT have like thousands and tens of thousands of dimensions, which lead to billions of parameters, and we can map out different things within that space. And the space is diverse enough to actually, code for the human language. So that's like dimensions there. And those are Euclidean dimensions. There's something else, they're called fractional dimensions. So they are fractional spaces. So for instance, when you're talking about a square, you can present a square in a 2-dimensional space. You can present a cube in a 3-dimensional space. There are things like, for example, a cauliflower, or for example, the coast of British coastline, the coast of England. When you look at that coast, the coast is not constant. You cannot measure it. Why you cannot measure it? Because the measurement itself depends on your measuring device. So if you have a scale which is like, let's say 1 mile long, your final output relates to 1 mile. The variation is in the scale of 1 mile. What happens if you increase the scaling? So which means you reduce the scale, you have like a 1 meter scale, and suddenly the coastline becomes bigger. You go to a 1 centimeter scale, the coastline will become even bigger. Why? Because you're able to measure things which are beyond that scale. So as you reduce your scaling, or you increase the number of scales you can account for, the bigger is the coastline. So in real terms, you can never measure accurately the coastline of the Britain because it has like a scale invariant relationship with your scale. Now the same thing happens in behavior. Normally, when we look at discrete behaviors, we are looking at one particular scale. But when you actually look at fluctuations or behavior, those fluctuations also have the similar property. So for instance, when you're looking at somebody is walking, when you look at the stride, if you look at one stride, there's something. When you look at 10 different strides, you see a kind of variation. When you're looking at 100 strides to 1,000 strides or 10,000 strides or 100,000 strides or 1 million strides that a person takes within a year, the patterns are very self-similar with a certain variation across all these scalings, which means that when you look at 1 million strides, you see certain things which you cannot see value you're looking at, like one individual stride. So why multi-scaling? Because we are looking at all these temporal scales in the case of strides, for instance. So when a typical biomechanist model a gait cycle or a gait, human gait, they model one gait cycle and they take the average of all the gait cycles. When we model the gait cycles, we do not take the average of all gait cycles because they are non-ergodic. And I'll explain what non-ergodicity means, at some point during the presentation. So we model the whole gait, all the gait cycle, because each gait cycle is connected to each other gait cycle, and we can statistically show that. So for instance, when you start walking, your first step and your ninth step is not independent. The way if you have to walk a certain distance, you have signatures of your ninth step in your first step itself. And that's what we look for. What happens is, over disease, for instance, the relationship deteriorates or it changes. So we can diagnose diseases, we can come up with biomarkers, look at this relationship and things of that sort. So that's the primary interest of the lab. The secondary interest is more in the epistemology of science, especially sensorimotor neuroscience. And that's what this presentation is focused on. You'll see that the title is Rethinking Human Sensorimotor Performance Beyond Engineering Assumptions and Towards Fluid Understanding. It's a little radical take, given that the dominant assumptions in neuroscience are coming up from our ideas of engineering or cybernetics, and still they are related to that some way or the other. And what we are trying to propose is that we need more like fluid dynamic understanding of these things. A very simple argument is that your brain, if you think about it, what your brain is composed of, it is fats, proteins, and carbohydrates. The brain resembles in composition more to a milkshake than it resembles to a semiconductor. But when you look at how we understand brain, we think about in terms of neural processing, we talk about error signals, we talk about representations, which are very metaphors related to the computational assumptions. When actually you're talking, the real question is that how is that milkshake is able to behave like a computer? Because ultimately it's fat, carbs, and proteins. And there's some kind of interactions happening within fats, carbs, and proteins in a very fluid fashion. It's fluid, first of all, which is leading to these kind of adaptive behavior. So we use techniques from fluid dynamics to see how these things are interacting. People have used these fluid dynamic techniques to study, for instance, ion channels, to study the flow of different hormones in the body, to study blood flow. We particularly use it for movements. And we are trying to propose these alternative models beyond current assumptions. So in this presentation, my objective is to show you what exactly are the assumptions of the engineering of the current dominant framework, and what our experiment shows or contradicts those assumptions, how, and what are the new assumptions which we propose. So me and my colleagues, we have these revised assumptions, and most of our papers and work is kind of a revisionist and radical in that sense to promote these alternative ways. Now, I know that most of you have been trained in general in representations and representations as the basis of cognition. So some of these things which will be a little novel to you. So take whatever you feel legit, and leave the rest. We all have our different takes on different things, so that's completely fine. So when we talk about sensory motor performance, there's a critical perspective of sensory motor performance, which is almost like a century old now, or 3/4 of a century. It started with a Soviet physiologist called Nikolai Bernstein. So Bernstein was appointed by the USSR and he was a scientist, physiologist, to increase the productivity of labor. And he was interested in how labor movements work, how we can increase the productivity of an average factory worker. And Bernstein was also the first person probably in history who started doing motion tracking of human movements. So this is a old setup from Bernstein's time, where he's trying to study people who are using a hammer to hit on a nail. And they used to put these different markers on the body and then look for, using very crude cameras, they used to take pictures again and again and see how the body was changing and track the movement. And Bernstein was, fascinated by this question of how to find the optimal sensorimotor solution. Now, if you look for the human hand, and human hand has multiple degrees of freedom, right? We have multiple joints. So we can create the same hammer trajectory each and every time, again and again, in different ways, so that your end of the hammer is always the same, but you have a different motion of the body, which is creating that particular end trajectory of the hammer. And this was very surprising for Bernstein because he couldn't figure out like how is that the case. And he posed this question that the big question is how the brain selects one solution over the other. And it's called the optimal sensorimotor solution. Now, optimal is a very important word because most of engineering basically is the whole engineering enterprise is about optimality, about optimizing things, right? And the same way we think about the brain. So if brain has 5 different ways, how does it choose one over the other? And we come up with some function, we'll optimize some parameter, and we'll say based on these factors or this input, brain is optimizing this particular quantity, which is associated with this particular movement, right? So this was the issue that you can see that there are multiple trajectories, each trajectory are different, but still the hammer strikes the same point. So Bernstein created these plots. He posed this problem. And later on, when he started pondering over this problem, it turns out that it's not just a joint space in which there are multiple degrees of freedom. When you think of muscles, you can change your leg in a certain fashion by recruiting different kinds of muscles. You can do it by recruiting the same muscles to different levels. When you're talking about a muscle, you can recruit the same muscles based on different neural innervations, right? So as you go from the level of the joint space to the muscle space, to the neural space, and then to the neurons in the brain, you have much and much higher dimension space, right? The space becomes more and more higher dimensional. It becomes almost intractable. And now the question is that, how do you coordinate all of that infinite number of degrees of freedom to produce a single movement? So the belief currently in neuroscience and sensory neuroscience generally is that the human movement has an optimal solution to this redundancy. And people have written a lot about it. So one of the quotes, for instance, capturing that fact is, the optimal policy has a property that whatever the initial state and the initial decisions are, the remaining decisions must constitute an optimal policy with regard to the state resulting from the first decision, right? So you already have these precursors to prospective nature of your first initial stride or first initial stuff in, yeah. So I just have a question from someone that muscles, joints, brains, neurons are the different scales. Okay. So I would say they are at different levels and they also at different scales. You can think of scale in terms of space. So for instance, I personally associate these at different time scales. So for instance, your neuron is firing, at like several thousand hertz, right? Then you have your neural networks, which show activity at much smaller scales, much larger scales. Your muscles have even higher scales of activity, and your joint is moving at the scale of, for instance, 1 second. If a movement, takes 1 second to, unfold, your joint is moving at 1 second. Your muscles are firing much faster. Your neurons are firing much, much faster. And when you look at the actin and myosin filaments inside that neuron, that is firing at even faster scale. Now, when you look at muscle, the whole muscle is moving at a slower scale. But when you look at different muscle segments, and again, you have actin and myosin filaments in the muscles, they have to act even faster. Now, when you look at actin myosin filaments, they don't move like in vacuum, right? They have to have an energy. So you have a constant generation of ATP, for instance, inside the muscle. Metabolism is going on. That metabolism is having even much finer scale of activity, very, very fast, at very, very fast movements. Now, the challenge for the body is to coordinate these activities which are happening at a very molecular scale, at a very fast time scale, compared to this whole body movements which are happening at very small scale, and they are all coordinated. So the challenge is that how do you come up with this control? If you think about brain controlling, using some signals, using like an internal model or planning things, then it becomes intractable because what exactly has to be optimized, what exactly has to be tracked, what exactly has to be corrected, we still do not understand. They're all speculations. So this is where the scaling comes in. Yes, space scales and time scales. So it will be more clear, as we move and talk about time scales. A lot started with cybernetics, with the work of Norbert Wiener. where he developed models, of cybernetic models of language, cybernetic models of movement. And when you look at early movement work and solutions to Bernstein's problems, people come up with internal models or different kind of models. They were taking it feedback, and one of the prime examples is what governor, in certain ways, that you have some kind of a central tendency of the system, or you have a monitoring device somewhere in the system which monitors, the whole system, corrects it, and there's a constant feedback. There's several problems to this. For instance, you can ask, when we are looking at any feedback-based model for the brain, where is the feedback arising? Is feedback packaged? Like, for instance, when you're talking about computer, it is parceled in small chunks and the equidistant time. In the body, we do not have any central clock. So how does the brain synchronize different error signals from different parts of the body, for instance? They are not parcelized at equal time. So if they're not parcelized, how are they, even able to compare or across different kind of error signals? So there are a lot of gaps between cybernetic and, computational explanations of how these movements are controlled. Again, since we do not understand this, it's open to abuse by a lot of mathematics. So we have a lot more complicated models now. And now we also have machine learning and artificial inside that. But let's come back to assumptions. So what are the assumptions which have lead to, that kind of framework or that particular restricted understanding of the body? So the first assumption, generally speaking, is that the body is basically bones, is a skeleton of bones, which serve as a rigid framework. And on that bones, you have muscles which are attached to it, right? And your bones and the muscles allow to move that bones like a lever. So for instance, if I move my elbow, right, the muscles of my hand, they act as a lever. So based on what force I produce, I'm able to lift my hand because my elbow joint is a lever, right, is a fulcrum. Likewise, the shoulder is a fulcrum to the muscles attached therein. And likewise, your knee is a fulcrum for your, lower leg and upper leg muscles. And we can explain those based on different kind of levers. For instance, the first class lever, second class lever, and third class lever. That's an assumption, right? It is coming up again from an engineering perspective. So when you look at a leaning tower of Pisa, the leaning tower of Pisa is made of one thing built on the top of the other thing, right? So you have one floor on top of the other floor. That system is gravity dependent. So for instance, the leaning tower is leaning, and that's why it is famous, right? Because it's leaning and still not has fallen. It leans a little bit more, it will fall, right? There's no way it cannot fall. Because the structural integrity of the upper, of the lower depends on how the upper ones are shaped, and the upper ones are related to the lower in a particular linear fashion, right? When people were building Leaning Tower of Pisa, they did not build it, flat on the ground and then they lifted it. They built one story on top of the other, right? There was a directionality, a gravity dependence on that. We assume that the body is the same. But when you look at the body, for instance, we can do all sorts of behavior with the body, right? Body has no directional specificity. Yes, our vestibular senses are attuned to the direction of gravity, but at the same time, body has no issue about its structural integrity in terms of standing, for instance, upside down, right? The same person can, put on weight like 100 more pounds, and the body is still able to do things, right? The structure is still intact. In fact, the bone loading forces increase and bone becomes thicker, as you become fatter. So the body doesn't follow the linearity principles, which we started with, from engineering. And we can do all sorts of things with the body. For instance, I can go to an extent that telling that the idea of center of mass is kind of flawed in the body, because static objects are center of mass, but we can control a body in different ways despite being center of mass, being away from the base of support. So the same Newtonian mechanics which works in Leaning Tower of Pisa doesn't work with the body. When you look at how the structures are, engineered in the real world, we have a step-by-step incremental process of building, right? We start from one story to the next, second story to the third story, and things like that, right? There's never you start with a small building and somehow, blow air into that building and make it bigger, right? We always start from one end to the next end. Whether you're talking about 3D printing, like almost every manufacturing process which humans have made starts from one particular dimension and moves to the other dimensions in space. When you look at the human body doesn't develop like that. The human body is a whole to begin with, and everything develops at the same time, right? no other part is dependent in terms of direction to the other part of the other part of the body, and it is gravity independent. So if you think about a human, baby inside the womb of a mother, the mother is doing all sorts of things, including acrobatics, handstands and everything. And it does not influence the baby's development because the baby's growth is independent of the gravity, right? It happens in all dimensions. It does not happen across like 1 or 2 dimensions. And it happens all at the same time while the structural integrity of the body is all combined, okay? Same thing happens when the baby comes out and starts growing. So that's a first assumption, which is not valid for the human body, right? Second assumption, which is based on engineering principles and determining most of our neuroscience, is the idea that you can decompose the system into independent components and you can study them in isolation. So typically, when we design an experiment, we try to isolate. For instance, when you look at early papers of reaction time, we divide reaction time into different components, right? For instance, a signal going into the brain, right? Then, brain doing the processing to make a decision, for instance. So you can divide things into chunks, and once you delete or you incorporate another task, you assume that there'll be some addition to the time or to the performance, and you can come up with like statistically different factors, which again have additive interactions with each other in your statistical model. And the idea is that if you can account for those factors in additive fashion, finally you can add them together to form the body or to form any cognitive process. Now, when you look at typical brain maps, they look like this, right? Where things go from one side to the other. There are linear routes to processing, and different parts of the brain are, interacting in a very linear fashion to different parts and things like that. The idea is that somehow we are able to change the topology of the brain. So think of the globe, right? When we put a 3D globe on a 1D map, right? There's no map which is accurate. In cartography, it's like an untractable problem. There's no map of the world on a 1D space which is actually accurate, because it's impossible to map accurately a 3D globe in a 2D space, because topology doesn't allow it. Topological theorems make it invalid. Now we are trying to present an infinite dimension brain in a 2D space with lines, right? So the same thing happens. There's a cartographic problem where we don't understand, first of all, what exactly we are mapping. So when we say that something is a node, we assume it's a node. It might not be anything like node, because in the brain there's no specific region. Everything is like blurry, or where a region starts and where it ends, and other than like some specific lobes, there's no specific boundary, right? But we have divided based on functions, and then we try to look for linear interactions among those based on whatever divisional experiments we have done, which is better than nothing, but I'm trying to show the limitation here. So the assumption is that just like a car, if we can take each part out or knock each part out at a time, we can identify its function. And if we can combine that together, we can make the car once again, right? So each part has a specific function in a car in itself. So for instance, a tire, even if you take out the tire, it still can roll, for instance. And that rolling, when you attach to the car, it allows the car to move, right? So there's a function independent of the car, and there's a function which adds to the car. Likewise, the seats. We can sit on the seats no matter whether the car seats are inside the car or not. So there's a function of the seat. When you put that seat inside the car, it allows people in the car to sit, okay, in a particular fashion. Same with the door, right? So each of these quantities have their own identity, and when they're combined in certain ways, they make the car, as an entity, with each of them preserving their own identity while they're also the part of the car. That's the assumption. When you look at the human body, most of the things are so interacted with each other that you cannot even take them out. For those, like, if you don't believe that, I really urge you to take an anatomy class or do a human cadaver dissection. When you do a cadaver dissection, you realize that how much more complex the human body is, or the brain is, than we actually attribute it to, for instance, and the central nervous system and the innervations and things like that. So no part of the body has any functional integrity, or even existence when you take it out of the body, right, in isolation. And it is extremely related to each and every part of the body. The body just cannot function in isolation of any of those. You remove the seat of the car, it still works. You remove the nervous system, the human body will not work, right? or you remove like blood circulation, human body will not work. That's not the case with the car. The car will still have some identity without having some, some particular part of the system. So human body is different. That's assumption two. The third assumption is that the central nervous system is representational, which means it is capable of representing the outside world. It senses and it predicts the outside world. So for instance, typically, like you can see this example, that the image of the dog in the person is going in the brain and somehow it is being registered and we are making a representation of it. And the picture of that percept of that dog is coded in neurons in certain fashion, right? That's a representation assumption. Now, I'm not denying the fact that we make representations. We do make representations. I am questioning the assumption that their brain works in a representational fashion. There's a distinction between the central nervous system working in a representational fashion versus it having representations. They're two different things. So, so now when you look at a semiconductor actually has representations, right? So you can actually look at the state of a particular transistor and you can map those state and you can identify. And there are experiments which have been done, for instance, by Conrad Cording's group, where they gave semiconductors to people to identify what games were actually stored. And to a certain extent, people could find just by breaking down semiconductors what was the nature of game, within that. But it was a failure still in a large part. So even semiconductor is much more complex, than we typically attribute to in terms of representations. When you're looking at a human brain or you're looking at neurons, you have no two neurons which are identical. Okay. Each of the neurons have thousands of input to thousands of other neurons, right? We have tens of kinds of neurons. The neurons are different in shape. The neurons are different in length. They have differential lengths of their dendrites. Now we know, at least from our physical models, that each of these factors affect the speed of conduction in a neuron, the strength of conduction in a neuron, and we still do not have any idea to map interactions between 10 different neurons, because just 10 neurons connected to each other will create a complexity which is computationally intractable, right? So when you're looking at C. elegans model, for instance, or Hydra model, where we have mapped the whole neural net, we have basically mapped the connections. We have not mapped, each and every impulse and its conduction there. So a neuron is much more complex to represent anything. And if it represents, there's no physical connotation or idea we have that how it is actually retrieved. Okay, so I have a question that, about the epistemology of modeling, right? It is true that you cannot have a model, and that to completely model the brain, you need to basically create the other brain, right? Like the perfect model is to build the same thing which you are trying to model, right? But at the same time, some models are more correct than other models. So I'm not trying to say here that we need to simulate neurotransmitter being signal. I'm just trying to go towards more accurate modeling of the brain than less accurate modeling of the brain, at least. So it's incremental. I'm trying to propose an incremental, improvement here. But
Madhur,
you're right. Yeah.
can I just, we're a small group, and so Frauke and anyone, feel free to speak, to ask questions. We don't have to write messages. So please, Madhur, I have a question, if I may, and that
Yeah.
concerns what you said. You said there's a difference between the brain having representations and something, using
And
them, acting.
operating on representations.
Yeah, would you, I don't know if you're planning to go back to that, but I would like to hear what that distinction means in your mind.
Okay. So the distinction is that, so for instance, if you ask me, do I remember, my parents' face, for instance, right? Now, my parents' face don't exist right now in front of me, but I still have a caricature of that inside my head, right? So yes, we have represented what is outside the world. We can model that inside the brain. Yes. So I'm not denying that fact that we can model the outside world. But I'm denying the possibility that the brain is operating on those representations. So for instance, all our digital models or all our computational models, we represent things in a certain way. It might be, for instance, um, a particular spike train, the way we kind of now, attribute representations to a structure of a spike train, for instance, right? I'm denying the fact that it can happen like that. We can have representations, but we still do not understand how those representations are formed. So we don't operate on representations or representations of visual information, for instance, but we do create models by things, by doing something which we do not yet understand.
We scientists do not yet understand.
Yes.
Um,
I mean, of course, people have different ways. So, for instance, I have a simple question which I asked to one of the people. So, for instance, Nico Hatsopoulos, who does ensemble recording from monkeys, and they encode like reaching movements. I asked him. And they the way they treat neural spike trains is that neural spike trains encode movements or represent movements, right? A particular spike train. Now you have people now who have used alternate technology, for instance, calcium signaling, where you put calcium dyes in the brain, or you look at just calcium activity in neurons, and you can record it by a light signal, and you have this neural, basically instead of spikes, you are recording the light activity through that calcium signaling by attaching a fluorescent dye, and you can decode that as neural activity. Now, there is no one-to-one correspondence between neural firing and that activity. It is not. But at the same time, you can decode or you can relate, using your algorithms, movement to calcium signaling activity. Now, the thing is, that questions, do spike trains actually encode the movement? Or is it the chemical activity which encodes the movement? Or is it something else? My proposition is that, which is again radical, is that we came to believe that spike trains represent something because we had the electrode to put in the brain first. If we had come with the light techniques first and electrodes would have come like later in 22nd century, we would have thought that it is actually the calcium signaling which is representing things. So our ideas are very much dependent on what the dominant technology to probe the brain is, rather than actually having a rooted explanation. When you will talk to more radical people, like you will talk to, for instance, Stuart Hameroff, he will talk about actin and myosin activity in the neurons, which is fluctuating at a much higher frequency at megahertz. And actually, there's a lot of evidence that the states of actin and myosin filaments in the neuron can do much more computation than what neurons can explain, or can have much higher dimension. So we do not yet understand what exactly happens and what exactly is mapping the outside world. But we do know that outside world is mapped in the brain.
So we should not confuse various indices with the state to be explained.
Yes.
Thank you. Okay, that helps. Thank you so much. Okay.
So that's the third assumption. And there's a fourth assumption. The fourth assumption is that the physiology and performance are ergodic. And because they're ergodic, they allow us to generalize our experiments across, events, across people, across conditions. So this is a very important concept. We can go back in history, and I mean, I can talk like 5 hours on this topic of ergodicity, my favorite topic these days. So when you trace back the history of thermodynamics and almost every statistic, our forefathers who started the field of thermodynamics, they encountered a problem. And the problem was that when you're talking about a gas molecule in like a box, and you come up with gas laws, for instance, or thermodynamical laws, they encountered that not every molecule is likely to travel every space inside the box. And that was a big problem because there were unknown number of molecules in the box. So how do you derive the thermodynamical equations? So they started this assumption. The assumption is that every molecule will travel outside all sides of the box, and we'll assume that we will look at the box to an infinite time, and given an infinite time, just like a monkey can type a Bible, if given that infinite keystrokes, by chance, the same way, you have every molecule traversing every portion of that box if you look at for infinite amount of time. And that assumption went to the thermodynamics, and all our modern statistics is based on the assumption that we can look for n number of individuals and talk about time. So for instance, in this case, I show you different graphs. Each graph is a time series, and there's a realization of this time series of different processes. So the assumption is that that if you have one time series and you average that time series across infinite time, we call it a time average. And if you have, n number of those realizations and you take a sample across space, right, across that ensemble. So you have 100 participants, assume you measure 100 participants one time, or you measure one participant 100 times, you will get the same value. That's the assumption on which all our modern statistics is based on. All Fisherian statistics is based on this. So, and that's how we design our experiments. So for instance, sometimes we have 500 people in a clinical trial, and we assume that 500 people will reflect the behavior of every person, over time. Or some studies, look for people across the lifespan to understand how everyone will behave. But the idea remains the same, that we can either look in space or look in time, and we can generalize across these two dimensions. When we can do that, that's called ergodicity. So when a measurement can be generalized, it's called an ergodic measurement, and the system is called an ergodic system. By the way, only dead systems are ergodic. Every live system is non-ergodic. Like how? So you can look at a coin toss. So on the top left, you have a simple example of a coin toss. So you toss a coin, you either get heads or you either get tails. It's a perfect example. And when you look at the time or space, average, you can toss 100 coins. 50 coins will land, if they are not biased, as heads. 50 will, tails, right? And you get 50-50 proportion. Alternatively, you can toss the same coin 50 times, 100 times, and you assume that 50 times it will turn head, 50 times it will turn, tails. So you can generalize, you can either take one coin 100 times or 100 coin one times, it doesn't matter. You can call 100 people, toss the coin once, or you ask the same person toss the coin 100 times. It does not matter. It's an ergodic system. When you go to the roots of our statistics, all our statistics is based on this assumption. It's frequency-based statistics, right? Every frequentist statistics is based on this assumption. When you look at any process, which is a little more historical, let's talk about first order autoregressive process on the top right system. When the system starts, it shows a lot more variance, but after some time, the variance reduces. And then you can, generalize across that system, whether you're looking at space or time. But as soon as you go to a little more complexity, so for instance, in the bottom one, you have a pot, you have two balls, blue and red. You take one ball out, whatever color you get, you put an additional ball. Then you take another ball out, and then whatever color you get, you put another ball. So what happens is that over time, your first move will bias, or first 2-3 moves will bias, and the trajectories deviate, as you can see on the bottom right, from their initial trajectory. In these cases, when you take the average of any particular trajectory in space, in time, sorry, and you take the ensemble or average of multiple such trajectories, you cannot generalize from one to the other. Okay.
Can you say that again? Because you're going very fast, and this is very deep. Can you just go through that one example again very slowly? What are you plotting on the bottom right exactly?
So on the bottom left, you have an experiment. No, none of them is Brownian motion. To answer the question, so on the bottom left, you have a pot and you have two balls in it. One is red and one is green.
Yeah.
You put your hand in, you take out a ball, and whichever color you get, you put one additional ball of the same color. Then you do it again.
Of the same color. I missed.
Yes.
I didn't hear you say that.
Yeah.
Okay.
So you keep doing that again and again and again, and you will see that at the end, the probability of a green ball will diverge from its initial condition based on some initial moves. To an extent, the true trajectories might completely different. So two people might do the same experiment parallelly, but they both will reach different probabilities over time, and the probabilities will converge towards 1 or 0 or very little, over time. So this is a process which has an autoregressive component, a higher order autoregressive component, basically initial state determining the future state with some probability, with some fixed probability. In this case, a moving fixed probability based on the total number of balls. And then if you find a time average of any particular trajectory, or you find an ensemble average of multiple trajectories, they will never be the same, right? The system has broken ergodicity. That's what it is called. The system has broken ergodicity. And this is a very simple process in which the balls are fixed. You can have a system in which somebody else is placing number of balls in it, which you don't know. There can be n number of variations, right? Yes, it can be represented by a Markov process. It can be approximated by a Markov process. This particular example, but not more continuous examples. So, so now the thing is that when you look at physiology, most systems are even more complex, and then they break physiology even more. So the idea is that your Fisherian statistics, your frequentist statistics, or the assumption that you can generalize across space and time is only applicable to top left system and top right system, after they have stabilized. When a system is not in equilibrium in general, which every system is when you're talking about a human physiology, you cannot generalize. So for instance, you scan a person, and every time you will do the same stimulus, the previous stimulus will influence a little bit more, or in certain ways, if you are able to look at particular scale. So so basically there's a time dependence, there's a history to every physiological process which influences, the future. When you look at the computational models of the brain or in general of physiology, none of that account for this kind of non-ergodicity. In fact, all machine learning and all AI right now has no ability to do this, and we are doing parallel work to show that when the system has a broken ergodicity, we do not yet have even artificial neural networks which can deal with it. They just behave very abruptly. So,
Could I ask
but again,
another question? I'm still, this is all very new stuff for me, and
yeah,
I'm still thinking about the simple little probability game where you pull out a ball and then you put it back and then put in another ball of the same color,
yeah,
and grows and grows, right?
yeah.
Okay. And I get that the exact outcome will depend on the history, but averaging over all those experiments will yield something very sensible. It's a little bit like the difference between predicting the weather versus predicting the climate,
Uh, okay, so predicting the weather versus predicting the climate.
which is
Uh,
very local in time predictions versus some kind of averaging over time. That's what weather is. Sorry,
uh,
climate. That's what climate is rather than weather.
yeah, but
If
the
you
thing
do
is
this,
that,
the balls experiment, you do that same experiment 1,000
yeah,
times and average,
yeah,
it's going to conform exactly to
yes, yes,
standard classical
it will,
probability theory.
yes, but it will not tell any information about any one particular probability. So, for instance, you can run an experiment and you do it 100, 1,000 times, million times, whatever, and you will find an average which will be like about 0.5. Yes, but that 0.5 will not reflect any individual in that population. When we can simulate this, so your population will be on two different sides, and you will find a mean, but the mean will not reflect your any individual there. So your mean will converge. Yes, your mean will converge over time, but the mean will not represent any specific individual trajectory.
I
But
see
the
the
thing is that,
problem. Now I see the
right?
problem.
The thing
And
is that
by
when we,
analogy to movement, you want to be able to predict
yes,
individual movements
yes,
or
there,
properties of them. Okay.
there's a reason why we have tens of thousands of studies which look at heart rate variability and publish a paper, but we still do not have a heart rate variability based biomarker for any disease. There's a reason for that, because we are still stuck with statistics which do not account for this property of heart rate, and we apply all that statistics, throw up all the computational power, but we still do not have a heart rate variability biomarker
Okay,
after tens of
Madhur,
thousands of it.
I'm with
Yeah.
you now. I'm with you now. I hope others
So,
are following. Every, everybody's following. Okay.
so that's a fourth assumption of dominant modeling frameworks. And this is another reason, for instance, we have decoded, for instance, a lot of brain activity in different tasks, but we still have less predictive power in terms of, what would be the brain activity for a certain task and for under what certain condition. Within the same model, within the same data set, we can predict. That's very hard to predict otherwise in general, or how an EEG will look like. When you look at artificial signals people generate, for EEG, for instance, those are very simple additive, autoregressive processes. They do not have the complexity which actually real EEGs. They look same because they are able to represent the mean, but they don't represent the complexity, which, it's very simple to, test for. So to summarize, so far we have 4 assumptions in engineering that won't serve the rigid framework, which do not happen in the real world, with the human bodies. The second is that you can decompose the movement system. The third is the central nervous system is representational, and the fourth is that physiology and performance are ergodic. These are 4 assumptions, basic assumptions on which most of our modern neuroscience has been, is based, and it works under certain conditions. We still get good results under certain conditions. When we look at a specific scale at which things are ergodic, when we look at a specific scale when things can be represented, when we look at a specific scale when things can be decomposed, yes, it works. And it also biases us in choosing those paradigms, choosing those, limited scope or that dimension space in which these assumptions hold to a certain extent.
May I add a
Yeah.
comment? Oh, and
Yeah.
by the way, as the host, I'm seeing that we are approaching the hour, and you have laid out problems with the engineering assumptions. I expect that part 2 will solve all these problems.
It won't solve, but it will give a little,
So-
hope. Yes.
And we could turn to that after the break. But I wonder if I may respectfully ask, with regard to these 4 assumptions, it strikes me that the only one that's really critical to the
Yeah.
argument that I think you're building concerns the ergodic process. Because if we think about the balls,
Yeah.
that does have rigid balls.
Yeah.
It is decomposable into independent components. And whether it's balls don't represent, it doesn't, that's not really. So the only one that really seems to matter formally is this business, the 4th one.
So you're right about it. Statistically, ergodicity is at the heart of things. So ergodicity is a reason why we cannot represent, why the brain cannot be representational, because our computers are actually ergodic. The brain is not. So ergodicity is the reason that you cannot operate on representations. Yes, we also have to talk about material composition and operations on the material composition. So ergodicity is more about, the change of the material composition. And the first 2 systems, okay. So the ability to not decompose contributes towards ergodicity. So assumption 2 actually contributes to assumption 4. So to the end, assumption 4 is the one which matters. The assumption 4 contributes to assumption 3 that non-ergodicity actually makes systems non-representational. Yes. And the first one is important because the first one is how non-ergodicity actually happens at the movement level, and how the body is able to solve things and compute or do things which resemble computation despite having things in non-ergodic. So first, the revisionist assumption of the first one actually helps relieve the problems of non-ergodicity in a human body.
I'm not I personally am not convinced by that argument because of the simple balls experiment.
Balls
Mm-hmm.
was a very simple example to show that things can break ergodicity.
Yeah. But I think there's a little slipperiness in the argument, just philosophically. I say it with respect that, the body is very squishy, and so and so ergodicity doesn't apply to squishy, messy, fluidic things. Maybe that's exactly what you do mean to say.
I mean, water is ergodic in terms of, molecular structure, for instance, it behaves, but at some scale, so it's the emergence in the body which leads to ergodicity breaking, and we have models for that. So for instance, when you are talking about additive systems, where 2 systems interact in an additive fashion or built on each other, they behave ergodically. When you have systems which have multiplicative interactions, and you can simulate these, using simple numbers, they actually break ergodicity. And it's a
Fascinating.
multiplicative,
Yeah.
interactive nature which creates ergodicity, which breaks ergodicity. And
Fascinating. Yeah.
so when you look at assumption one, when you're talking about bones building on each other and muscles just putting on top of each other, those are additive systems, and it should remain ergodic in terms of how it moves. And actually, when you
Mhm.
look at liver systems, they are ergodic in terms of the movement. But as soon as you're talking about a closed chain kinematics, which the body is, the same closed chain kinematic system does not behave ergodically. So
Okay.
it's a material composition which the body is, in a certain way that actually breaks ergodicity.
Okay. I'd like to suggest that we take a 2-minute break now because we've been going for a little over an hour. And Madhur, is this a convenient time for a 2-minute break and then we'll
Yeah,
come
sure,
back?
sure. Let's do this.
Okay. Thank you. Thank you. Great. Okay. See everyone
Bye.
in a couple of minutes.
Yeah.
Thank you. Okay. Brandon, I hope you're digging this talk. Madhur, I'm really enjoying this presentation. It's very stimulating. Very stimulating.
Thanks.
Really, very, very
Yeah.
stimulating. Yeah. Yeah. What's the name for this little probability game with the adding same color marbles? Does it have a name?
I don't know.
Okay, okay.
But it's like it's like 10 lines of code, and
Oh yeah,
you
I
can
could write it.
right?
I could write it in a second, but I'm
Yeah,
just in
yeah.
the literature, it's called the such and such problem. I was wondering if there's a name for it.
Wow. There's a lot of versions of, in probability. It's basically a second order,
Yeah.
order regressive process.
Yeah, yeah, yeah,
In
yeah,
terms of
yeah,
classification.
yeah. Yeah, I understand. I understand. Yeah. I just feels while we're waiting for this, it does feel like I still haven't, I've just never encountered it. And to me, it doesn't feel especially radical that the outcome depends on what happens at first.
No. So,
Uh,
so that is not radical because that is a very simple thing what I'm showing, and I'm not presenting the more advanced work. I'll circulate a paper to the group afterwards where we have cascade models and we can create multiplicative interactions and put a contamination of additive interactions and we can simulate almost every physiological process.
okay, okay, I see.
However, the problem is that you cannot code them. They cannot be resolved analytically. There's no analytical solution to them. You actually have to simulate every time, the problem with modeling.
Right,
So now the question
right,
is that when you cannot, show them using some, phase relationship or anything like that, then how do you actually model? How do you learn the properties of those? So then there are multi-scaling techniques which allow you to compress and look at certain feature across time, and no other techniques allows that. So
right.
the proposition is to adapt that statistics is called multifactorial statistics because that allows us to look at this kind of variation, and our previous statistics does not allow it to use. So
Okay, okay. Well, okay, yeah. So but you model the modeling that the brain does.
yeah,
The so the brain does Monte Carlo, and then we can use
I
Monte
don't
Carlo to simulate
I
Monte
don't
Carlo.
no, I don't. I'm not saying that brain does Monte Carlo. Neither I'm doing Monte Carlo. I don't think brain is like working.
Okay,
Brain,
I know. Just kidding.
if you think about a water flowing in a pipe, pressure changes, gradients happen, eddies form, right?
Yeah,
And they
yeah,
all have structures to them, and things turn. I mean,
yeah,
for
yeah.
instance, when you have when you have rainfall, you have cracks in the mud, and when it becomes moist, it comes together, forms a pattern. And when
Yeah,
it dries up at the same time, the whole of it dries up, and you have the cracks forming. So if you
yes.
think about Parkinson's, for instance, or any other disease, right? What changes in brain does not happen at like one instance and move. It everything changes at the same point, right?
I
It
understand
challenges
you.
it challenges the
And
whole assumption of how the integrity is
it's
maintained.
beautiful. It's beautiful. I'm with you. Please go on. Please go on.
Yeah.
People are coming back.
So I will show you now some evidence on which contradicts the first, in empirically, and then how we tackle that issue. So first assumption is that movement system is a body-wide tensegrity. That's a revised assumption that it's not a musculoskeletal system. It's a body-wide tensegrity structure, which has compression and tension elements, and they change their role, and it's a continuous system. So for instance, when you look at the body and when we put our joint, when you put our feet or when we bite, our jaws, we normally look for only acceleration and velocity in the labs. We are not concerned with jerk and snap and crackle. But you can actually look at derivatives of the forces and you keep on taking derivatives. It turns up that the snap and crackle is so high that any physical device will just crack into pieces. But the body is able to withstand all those big crackle and jerk and snap. Why? Because they are not big within the body. And why they're not big? Because body does not follow Newtonian mechanics. A body is a closed loop system. Here I'm showing you an icosahedron, which is a very simple system here as a poster child of what the system is. You have these elements which are under compression. So in this case, the sticks are under compression, and you have these threads which are under tension. And the structural integrity of the structure is maintained by the specific geometry of these two things. where one type of element is only under compression, one type of element is only under tension, right? This is not the case with human body for buildings, for instance. In the buildings, the beams are under tension and the pillars are under compression. And that's the reason when you change the direction of the building, it collapses. Why? Because the tension element is made to only sustain tension. Compression element is made to sustain tension in a particular direction. This direction is omnidirectional, this particular system. You can change it in any direction and it works, just like human body. So in this system, the force is spread simultaneously across the whole thing. When you apply like something on the wall, it does not spread to the other end. It just remains local. In the body, when you apply a force, it is distributed across the whole body because the body is a whole continuous system. And this can be tested in the lab. And that's the reason when we put a step, the impact goes even to the spine, to the head, it's all connected. And there's a specific geometry in which how it moves. So the way the structure is made, so in this case, I'm showing a very simple structure. Because it's a closed chain, it moves only, in a certain restrictor under certain constraints. And the idea is that those constraints themselves have smaller tensegrity structure within them. So no matter at what level you look at, there's actually a tensegrity system, working. So you have tensegrity building on tensegrity, building on tensegrity. There's a paper in Nature 2 days back. I'll forward you this paper after the talk. The paper has found the first ever molecule which actually is shaped like a fractal tensegrity. Okay, so we are discovering more and more of these structures in the body now. Donald Ingber at Harvard has, a lifetime career where he has shown that the cytoskeleton dynamics is not modulated by specific chemicals, but there's a lot of role in the mechanical structure, and those mechanical structures follow this tensegrity framework. He has like hundreds of publications showing this at the molecular level. We have very little evidence at the body level. So this tensegrity structure means that when you look at the liver systems, they are linear and Hookean. They have continuous compression. They are the gravity dependent. They have a unidirectional stability. They are rigid to be stable. They are open chain and they have high energetic cost. That's why robots get discharged in 5 minutes. When you're looking at Boston Dynamics robots, yeah, they look very nice. But when you look at how many times they are charged every day just to make that, 5 minutes of presentation, it's enormous. For humans, you eat like 2 energy bars basically, and you can, climb a mountain, right? We are very highly energy efficient systems. So when you look at the biological systems, they are non-linear and non-Hookean. Tensegrity systems are the same. We have continuous tension and discontinuous compression elements. We are gravity independent. We have omnidirectional stability. We are stable with flexible joints. We are closed chain. A simple example is hamstring, for instance. A hamstring is a closed chain, which connects your knee to the hip. And then we have very low energetic costs.
Madhur, Frauke,
Yeah.
I think it was Frauke, wanted to know what
So
non-Hookean
Hookean is basically
Robert Hooke.
Yeah, it's coming from Hooke's law. It talks about how systems behave, when you strain them. So for instance, you have like a cord, you have like some like a rod, and you stretch it, right? So the compression or load influences its behavior very linearly, and then suddenly it snaps. When you look at the body or the tensegrity structure, the more you apply the force, the more rigid it becomes. So you put on, for instance, you take your hand and you put like a thumb. It's very easy to initially press it, but the more you press, the harder it becomes to press because the system becomes more and more resilient with constraint. So the response is not linear. The response in this case is a polynomial function. That's non-Hookean. That's non-Newtonian. For Newtonian, F is equal to ma. For non-Newtonian, the A actually depends, it evolves in time. So the systems are non-Hookean. So we did a simple experiment, just to test this hypothesis in human perception, where we had these people standing on, force plates, and they were given different objects. They close their eyes and they have to wield them with different, frequencies, with different orientations of the hand, and estimate the length and heaviness of those objects. And they were standing on the force plate, so all we are measuring is the ground reaction forces, from the bottom. And what we found was that we looked at, and we also put markers on the body, and we did something called multifractal modeling, where we looked at the multi-scale interactions across different body parts and how those interactions evolved across time. So if the body is a Newtonian, and the hand is independent, we should see signatures of that perception only in the hand movements and nowhere else, right? However, if the body is a tensegrity, a closed-knit tensegrity, which is completely connected, then we should be able to predict perception of what happens at the hand and what the brain perceives from, any part of the body, because it will still have some signature because it is connected to the body. So we did this something called what vector autoregression modeling over multi-scale parameters, which we derived from time series analysis. And this is some of the output, where there are different markers, and we are looking at how putting an impact influences the response of different markers, in the time series. And here you have the results, for instance, for one participant. On the top, the heading is basically one marker's effect on the other. On the left-hand side, you have heaviness perception. On the right-hand side, you have length perception. Red means more effect and blue means less effect. So I want you to focus just on the plot, the bottom right plot. Basically, it shows the error. So blue means less error and red means more error. So blue means better perception. So now you are looking at the interaction between the hand and the center of posture, basically your ground reaction forces. What it is showing is that the people who had a much stronger interaction between their hand and their posture or forces below the body, they were actually having a much worse perception in this case. And when you look at, for instance,
What are these different panels? You're going a little quickly. What's the left and what's the right, and what are the rows?
Left is perception of heaviness, right is perception of length.
No, it's not perception. It's sorry, slow down. This is just measures of body positions.
No.
You're not asking people to give you verbal reports of what they perceive.
No, we actually give them so they wheel the rod for like 5 seconds, and then there's a pulley which they have to pull to explain to us, report how long they think the rod was, after
So slow down. Go back. Just go very slowly. When we talk about experiments, we want to.
So people.
What's the experiment? What was the procedure? What were the data and so forth?
People are blindfolded. Okay, they're standing on a force plate and they have markers on their body.
Okay.
So we record motion tracking, subtle fluctuations in the body, we record ground reaction forces. So every trial is 5 seconds, and there are different objects with different properties. They move it differently as per the trial.
Okay.
And we ask them, after they wheeled it for 5 seconds, and after 5 seconds, they have a pulley next to them, and they have to change the marker on the pulley to indicate how long they think the rod was.
Okay, as in Carrello and Turvey experiments.
Exactly.
Okay.
So what we found was that, and this plot is the result of the error in perception. So the lower means better perception, the bigger means worse perception on the y-axis. On the x-axis is the number of trials from 0 to 108. The left column are for heaviness and the right column are for length. And the 5 plots from top to bottom are the relationship between 5 different positions on the body itself. Yeah. Okay. So on the bottom right, you have the relationship between forearm and ground reaction forces. And what you see is that the bigger the impact of what happens on the forearm and posture, the more error in perception is over time. Okay, in the length perception. When you look at the plot, for instance, on the elbow to shoulder, the one on the right hand side, the second from the bottom, you will see the relationship reverses, right? The stronger the impact, the better the perception is. So we could find relationships within the body affecting each other, which were either good or detriment for perception. And we had very strong model in terms of predicting just from ground reaction forces how they would perceive the length or the heaviness. So what we showed during this work, that body is an integrated whole, and subtle fluctuations are connected in ways that resemble a continuous flow of mechanical perturbation, as well as this work is all analysis is done on multifractal. And multifractal is focusing on the multi-scale nature of those interactions across the body. That's what we are mapping. So we could find
what the red and blue line is. Is it important that they diverge in the bottom right?
So blue line is a strong effect, which means the stronger causal influence of one body part on the other. The red is the blue is a weaker effect. And what we are finding is that some body parts affect each other more strongly in terms of influencing perception, and they determine. So for perception to be accurate, you need to have less interaction between some parts of the body and more interactions between other part of the body. But overall, the interaction between parts is a crucial determinant of your posture, of your perception, of perception. So basically, we are providing evidence here of the integrated whole of the body, that you have signatures of what happened at the hand, below your feet, and we can model the whole as a network, which is
I'm can I ask you, is this published?
Yeah, it is published. I'll send you the.
But let me just play devil's advocate for a minute. This is very, very neat, very clever. But well, two reactions. One is you are back to looking at parts. And in a kind of standard regression way, saying some parts are more predictive than others.
No, I'm not making that case though. I'm not making that some part more predictive. I'm just trying to make the case that there's a coordination across the body and not just at the hand.
I understand. Well, we know that because every action has an opposite and equal reaction. This is all one system. Everything is linked.
Yeah.
And but still, just to play devil's advocate, I don't really believe this, Madhur, but let me just try it out for you, because we like to debate in science.
Yeah.
Devil's advocate. Well, people have learned to focus on just one body part.
Yeah.
Not take in information from all the different body parts. They just learn that, wow, if I pay attention to, a force feedback from the soles of my feet, that's B.F. Skinner would say the auto shaping in the pigeon learns to pay attention, so to speak, to that cue.
Yeah, but so in this case, people are actually attuning the perception to certain property of the object, which we can talk about inertia or torque or something like that. But how it is realized is not localized, right? The other thing is what goes into the model is not a linear relationship. This model is done over the difference between the nonlinear and linear aspects, which is what multifactorial is going at. So it is the specific information in the sequence of time, which is actually going into each of those points, then simple magnitude of interaction. So it is
a very simple additive model does much less worse.
We have already removed that. We have already removed that. So when we do multifactorial analysis, we create surrogate series which have all the additive components of our measurements, but do not have the multiplicative component. So it is only the difference between multiplicative and additive which goes into these models, and it is only that which is able to predict. If you insert the additive component, it has no predictive capacity. So that is called surrogate testing, and every of the a proper multifactorial analysis already has accounted for that as a part of surrogate analysis as a first step in terms of processing the data. So sorry about missing that, but
no, it's okay. No, you're giving an overview, and that's fine. Yeah.
So we published this on.
Aviad has a question.
Yeah.
Can you go back to the graph, please?
Yeah.
Is there, so I understand there is what's important here is, I hope I understand, the divergence between the two lines, right?
Yeah.
But what is the meaning of because I just can't see the y-axis. So higher values, it's just centimeters. I'm just
So higher value means more difference between the real and the perceived. So the higher the value, which means more error in perception, the lower the value is more accurate perception. So the most accurate perception means it should be 0.
Okay.
Yeah. So we published this in interface, and as our attempt to provide a more revised assumption that the body is not a simple lever-based system; it's a body-wide tensegrity. And since then, we have published multiple studies showing it in different contexts. And right now, we have a big project going on, NIH funded in the lab, which is also looking at the whole body-wide network. And we are trying to see whether, for instance, we have people with knee arthritis, for instance, and we are trying to see whether knee arthritis, in this particular instance, affects the probability of fall just like this, or it interacts with different parts of the body, and how we can, in rehabilitation, how we can, instead of focusing on a body part and rehabilitating that body part or targeting that, we can target the whole body, to do things. So there are different stuff after this published along the same lines. Second assumption was that you have multiplicative interactions across space and time scales, which do not allow decomposing using statistical frameworks. That's the second revision to the second assumption. So the assumption was that you can decompose, but now what you're proposing that you cannot decompose. So if you think about a simple example of a person, going to the office, right? If you try to model the movements, it's intractable because the person is thinking of going to the office, right? It has to, reach in time. That determines the gait cycle, the gait interactions that determine, the neural flows, and a little bit of perturbation can create a fall, which can change the future. So there are interactions happening, as I mentioned to begin with, from neural scale to the whole planning scale, from lower molecular interactions to the cognitive planning, okay? And your molecules right now are basically all working together for you to achieve that cognitive aim. And that cognitive aim is also constrained. For instance, you think of, a loved one and your heart rate goes up, right? So cognition carries a lot of importance for like lower order metabolism, and metabolism in turn influences, your cognition in a much longer scheme of things, just to, come back to that point again. So we did this very simple experiment where we had people standing and they had to look at a target, and we had different levels of different ranges. The target was at different places. And when you look at the angle of vision, basically, and sway, we have from a simple linear Newtonian model, as you increase the viewing distance, you would have a very simple relationship between what angle it basically forms on your eyes based on the viewing distance. And from that, we can, compare, how much the body should actually sway in time. Okay, so we have a Newtonian deterministic model of postural sway based on simple geometry. And we actually measured people's postural sway when they were doing that. And we did something called multi-scale probability density function analysis. It's a state-of-the-art technique, one of the most beautiful way to analyze this data or any time series data. So what it does is it takes your time series and it detrends it at different time scales. And then it looks at non-Gaussian behavior at different time scales. So normally most of our statistics assumes that things are Gaussian, right? And they fail under non-Gaussian assumptions, especially when we do not even know what the distribution is, whether it's log normal, whether it's exponential, things like that. This analysis lets us parse out our time series or behavior at different time scales, like 1 second, 2 seconds, 3 seconds, 4 seconds, depending on how you have measured. And you can look at a non-Gaussian behavior and how the non-Gaussian behavior itself changes across different time scales. So in this case, we did that. On the y-axis is the level of non-Gaussianity, that how much the time series breaks Gaussian assumption. No, it is not a Fourier analysis. So the brain is not a Fourier generator. Most of our analysis are based on frequency domain, right? Fourier analysis of frequency domain analysis again. There's nothing in the brain which represents a Fourier generator. Brain has no capacity to generate a sinusoidal wave. We have the capacity to filter the series and find sinusoidal waves based on a Fourier. And again, Fourier is an additive assumption. So Fourier basically assumes that you can make any time series based on adding sinusoidal waves, right? Fourier cannot deal with multiplicative interactions. It has additive assumptions built into it. Everything what we do is basically time domain analysis. So we don't go to the frequency because we do not assume that there are frequency bands. We just look at variability across time in different lengths of time. And there are pros and cons of both things. Both things lead to different things or different assumptions about the brain. But I normally say like brain is not a Fourier generator. That's my thing with Fourier.
And can I want to be of some service to the students here. When you use the word Gaussian,
yeah,
and that's easy to explain. Would you please take a moment to explain what that means for the
Gaussian
students?
is basically a normal distribution, right? Like a camel hump shape, where you have
a bell-shaped curve, guys.
A bell-shaped curve which has finite variance. The variance is finite. Okay.
Yeah.
In both directions. So Gaussian distribution has finite variance, and a non-Gaussian distribution has infinite variance, which means that you can have a time point which you cannot explain in terms of statistics. Okay. Yes, Gaussian also used for sometimes Gaussian noise. So here we are checking whether this noise is Gaussian or it is non-Gaussian. Okay.
And for the students, just so that for students, you may have heard, for example, that IQs follow a bell-shaped distribution, something
Yes.
like that. That's
so-
what Madhur is talking about.
I'll tell you, IQs follow a bell-shaped distribution, but wealth does not follow a bell-shaped distribution. Why? Because IQ is the way we define IQ is, some score in this, some score in this, you add scores in like 5 different things, right? An additive combination, you get a bell distribution. But the ability to make wealth in the real world, you need to look good, you need to be tall, you need to be educated, you need to be whatever, charisma, you need to be from a
Right.
well-off family. So all these interactions have to be there together. So then it's a
And-
multiplicative interaction. So you do
Right.
not have a normal distribution of wealth, and we will never have normal distribution of wealth, because wealth needs all the factors together interacting multiplicatively.
And Ari, you learned about Gauss. So it's named after Gauss, who
Yeah.
was a mathematician, brilliant mathematician in the 19th century.
Yeah, he was like a prodigy. So he used to solve. Read about his history, how he used to solve problems in school.
He was discovered, his teacher is responsible. His teacher identified him and recognized that this 8-year-old kid was very, very, very special. We owe a tremendous amount to the teacher of Carl Friedrich Gauss. Okay.
Yeah.
Sorry for the
And
I
he
just want to
discovered
try to
he-
bring everybody in. Here,
Yeah.
go ahead.
So in these plots, so a Gaussian distribution will have a lambda of 0, and the greater the lambda, the more non-Gaussian it is, right?
And what does lambda mean?
Lambda is a level of non-Gaussianity. It's a way to quantify non-Gaussianity. And I can forward a paper, talking about how it actually computes that. It basically looks at the peak. So your Gaussian distribution has a camel hump. The more non-Gaussian distribution becomes, the more tapered it becomes at the center. So very highly non-Gaussian distributions are like this, like a mountain peak, and they become flatter and flatter on the top. So lambda is a specific technique which doesn't look at the outliers. So our traditional statistics look at the outliers to identify non-Gaussianity. We look at the shape of the distribution. So it's a special technique where it can find things that do not exist just by looking even when they do not exist. So it's a way to predict future or uncertainty even when the uncertain event does not happen. So it's my favorite method right now.
And what was the word you used? Non-what?
Non-Gaussianity.
Virginity?
Gaussianity.
Oh, Gaussianity. Oh, okay.
Gaussianity.
The extent to which things are not perfectly
Gaussian. Yes.
bell-shaped, symmetric.
So we plotted the non-Gaussian nature of the time series with respect to the time scale here, the log time scale, in the logarithmic direction. And what we found was that, first of all, the time series are non-Gaussian, which the Newtonian model does not predict. And the second thing is that the non-Gaussianity changes with different time scales in a different manner. And when you look at the nature of the curve in yellow, the top middle, you will see everyone else is actually peaking up and down, but it has a very different kind of a profile of non-Gaussian interactions, right? This is also the distance, 25 centimeter, which is the most comfortable distance of viewing. So there's enough evidence from optometry, which talks about how 15 centimeter is the comfortable distance. So the top graph is basically the original time series, and the square ones in the bottom are the surrogate, which means we have removed all nonlinear interactions. We have removed multiplicative effects. The bottom one is a Fourier model, and the top one is actually the real time series. So the hypothesis is that the more you have to incorporate more number of processes, they will interact multiplicatively, and they will create more non-Gaussian behavior in your measurement. The less number of processes interact, you have less non-Gaussian behavior in general. What you find in this is when you close the eyes or when the object is too far, there's a certain time scale at which you find peak in non-Gaussianity. That peak corresponds to the time scale of different reflexes and voluntary contributions to the movement. So if things are additive, we will not see changes in non-Gaussian behavior, but if things are multiplicative, we will see changes in the non-Gaussianity of the behavior, right? Because additivity doesn't change the non-Gaussian behavior. And given that we know from past studies, what components go into posture at what time scales, for instance, certain stretch reflex go at 30 milliseconds, certain reflexes work at 50 to 100 milliseconds, voluntary motor adjustments in posture work at 200 milliseconds, we can trace the change in non-Gaussianity to a particular time scale, and we can talk about what new processes might have been recruited during a certain condition. And what we find is that, yeah.
I'm looking at the patterns here. You said that there's something special about the 25 centimeters, but it's kind of what's the unique pattern here? I think that if I look at the bottom, the non-original, it looks like it's purple one.
Okay. So there are two things. First, you should compare the most uncomfortable distance, which is when you have eyes closed and when you have eyes fixated at 305 centimeters, right? So the bottom right and the top left are the most non-Gaussian, right? Which is the most difficult conditions. The yellow one resembles a little bit like the top red, right? When you look at like visually, but when you actually model it, because we are looking only at the linear change, our eyes can only look at like one particular trend. But when you actually do the model, you look at linear, quadratic, cubic, and quartic changes in this. You will see that this time series has the minimal changes across all these four polynomials of the effect of time scale. So it is showing the least variation with respect to time scale on all those, cubic, quartic, and quadratic functions. So if you go to the original paper and you look at the results of the model, these are the mean results. When you look at the results of the model, you'll see that it cancels out most of the effects, showing that there's the least amount of influence of time scale on the non-Gaussian behavior of this particular condition. So from there, the conclusion is derived that it's the least effortful. But a bigger message to take from this plot is that from a normal assumption, green and purple are eyes fixated at two different distances, 135 centimeter and 220 centimeter. So the bigger conclusion to derive from this is that if you have additive interactions, you will not see changes in non-Gaussian behavior. And you will see that, when you look at the plots on the bottom one, all 6 plots, they are all very similar, right? Because they carry only the additive component of it. So you will see the same across all 6 conditions. But the top ones, the triangles, have different patterns across time scale because they carry the multiplicative interactions. Had there been only additive interactions, the 2 plots should converge. They will be the same overlap. But because it has multiplicative interactions, you do not see them. You have different profiles based on task constraints, which is an evidence that you cannot decompose, the behavior to additive components, but you need to have multiplicative interactions, and you need to count for multiplicative interactions for any conclusion you have to derive from this. So we basically had.
So Madhur, you need, therefore, just to go back, so you need some kind of super model that gets you through these different conditions to be able to explain the differences among those. Because you don't want to, you're not saying you have different regimes that set in for different distances.
Oh no, we have. So we basically have model, later on with different work where we can quantify those, using again multifactorial analysis, look at those interactions, and we have not yet gone to a level where we can knock down one of the system. That's my dream. So a
That's
future
the dream, right? Sure.
Where we will be knocking down different parts like reflexes or at particular time scale, and we'll show you in the same kind of plot in the spectrum that how knocking down that has a certain effect or not have a certain effect, and whether Fourier analysis is able to track those or not. We have not gone there because I'm just starting my position, my independent work, but we are going there, eventually. So we published this, as a revision to this particular framework, and it has a lot of implications. For example, a postural models are inverted pendulum models. where we think the body is an inverted pendulum, which again follows Newtonian laws. And we have more and more stuff coming in, and you'll see like several papers coming out from the lab in the next year or this later end year, where we will have a lot of proof about nonlinear interactions, multiplicativity, and non-Newtonian nature of the human postural system. Then we did several other experiments, along the same lines, where we show that when we change the task, it recruits more shorter timescale processes, and you'll see that effect in, again, non-Gaussian profiles. So that was it. Coming to assumption 3, the assumption
Madhur, could I ask may I ask a tutorial question
Yeah.
for me? Sorry.
Yeah.
What does non-Newtonian mean?
So Newtonian means linear relationship between force, and acceleration. Okay, think about this. Can you predict when you apply a force, to any part of the body? Can we predict, the response of the body in terms of using the Newtonian equation? We cannot. The Hooke's law is Newtonian. The effect or deformation is also non-Newtonian because Hooke's law ultimately is also
I see.
Of a Newtonian.
So you're using the word non-Newtonian in a very, gosh, it sounds very overblown. You're just saying if it's not exactly true that force equals mass times acceleration, then that's non-Newtonian.
No, non-Newtonian. I should use it less often. I agree with you.
Yeah,
I'm relating it to
the
more
universe, Newtonian mechanics works the same way forward and backwards in time. And,
It does.
Eddington
Yes.
and others said that's a problem. And to
It does,
say
yes.
if it's F equals MA doesn't quite, you need a non-linearity. And so that just feels, respectfully, I say, sounds a little overblown.
No, but I also agree with you that Newtonian also implies, reversibility.
He was a pretty
which
smart guy, that guy. So.
Of course. I mean, I'm not denying the fact at all.
Okay.
I mean, see, it's all built on the shoulders of giants.
Yeah.
There's no arrogance here. It's
Okay.
just an attempt to, get a little closer.
Shorthand. Yeah.
So the third assumption is the central nervous system is non-representational, right? And it is characterized by a power law cascading process. That's what we are proposing. So we have these gate models, and all the gate models are like that. We represent errors in signals, right? So we make something and we find an error, and based on there's error in the next round, we actually converge it to reduce the error. That's more internal models are, and we have internal models for walking. So we did this study where we asked people to walk on a track, and we had these people wearing these eyeglasses, and there was like a small LCD on the eyeglass, and it has a small bar, and that bar goes up and down, up and down, but it does not go up and down in a very simple manner, and goes up and down in different ways. So on the left hand side in PPS, it goes in a pink noise fashion, which means it has fractal property to it. The SPSS is basically the randomized signal, so it has no structure. The GRPS is basically a Gaussian signal, and the right one is a uniform signal. So we calculated the people's walking speed, and based on that we tuned the signal to give them predictable or unpredictable signal. Now, if the brain is representational and if it is correcting for errors, in that case the errors should be normally distributed, right? The error should be randomly variant across time, and we should not see a deterministic or a particular structure of their strides over time. But if it is non-representational, if it does not have an error signal, if it has some other way of working, then we will see something else. Then we will see the interactions and multiplicativity and things like that. So people have to time their steps to, when the bar reaches the bottom, and then the next step to the bar reaches the bottom. So basically they have to look at this and predict, the bar and time it. And there's an error, anticipatory error, like whether the step is after the bar or before the bar and things like that. So in this case, what you're seeing is different signals. On the top is the real signal, which we give them. On the middle, you have their stride interval, the interval between one stride to the next. And the left is like the real walking signal, and on the other four are the time when they have this cue presented to them. In the bottom is the asynchrony, which means that how much is the difference between their step and the real signal. And we look at the structure of error and structure of their strides across the structure of these four signals. This is ergodicity breaking. So far, we have only one measure of ergodicity breaking in the statistical physics community. It's called the ergodicity breaking factor. So on the y-axis, you have something called ergodicity breaking factor in this case, and on the x-axis is time. So ideally, a signal is non-ergodic, is ergodic if EB basically converges to 0. So the gray lines you see, right? All the gray lines are basically ergodic, and all the colored lines are basically non-ergodic if they deviate from the gray line. If they deviate, the more they deviate, the more they break ergodicity, which means the more historical the process is. So you look at in the top plots, the PPS signal is very ergodic signal, right? Is non-ergodic signal. It breaks ergodicity. But SPSS, GRPS, and URPS, all these three signals are ergodic, which means they're random. Okay. When you look at the stride intervals,
Ari has her hand up. Ari?
Hi, sorry, I don't mean to interrupt. I just have a quick question. So
Yeah.
this is for assumption 3, right?
yes, yeah,
Okay. And this I'm just asking because I just need clarification for myself. And you might get to it, but my question is that I saw in assumption 3, it says that the assumption is that it's not based on non-representational,
yeah,
but kind of like physiological
yeah.
power law driven processes. So based on this experiment, I know you're probably going to get to that, but I'm kind of confused on how this
Yeah. So
leads to
we
how
tackled
this
only we tackled only
data
one
leads
part
to
of
this
the.
assumption.
Yeah. So non-representational has or
Maybe I'm like
representation has.
rushing asking, but I'm just a little I'm struggling to find the connection.
No, I
No, it's
think
fine.
you're not alone, Ari. Thanks for asking.
So non-representational, so representational models, representational
Okay.
computational models have multiple components to that. One of the components is the error signal. Okay. In this experiment, we are only tackling the error signal. So and we're looking at the distribution of the error signal, and the hypothesis can parsimoniously explain whether the error signals adhere to the computational model or to non-computational cascading model. That's the only thing we are checking with this experiment. Okay.
Okay. Got
Okay.
it.
So see, these assumptions are very wide. An experiment is a very unique, focused on one simple thing, right?
Mhm.
And you for the whole assumption, you need multiple experiments tackling multiple of those, components of that assumption. So in this case, only one assumption, one particular component. So it deals with error signal. And what we do is that you have a participant, for instance, you will see in the blue curve is very close to the gray one, which means the gate cycle of that participant in this case is ergodic, basically random variation. And we artificially, perturb basically gate by asking them to coordinate their gate with different signals. The first signal is non-ergodic. The last three signals are ergodic because the two plots converge. When you look at the gate cycle or the stride interval sequence, when the signal is non-ergodic in the PPS condition, you see non-ergodic behavior emerging. And even when we randomize that PPS signal, you still see some kind of ergodicity breaking, but not for the random signal for the GPS and URPS. But when you look at the bottom, the asynchrony, the error signal, the sequence of error signal, you see very strong ergodicity breaking of error signal for the first two conditions, and also the ergodicity breaking of error signal for the next two conditions. So even when the Qs were completely uniform in the right-hand curve, the green one, and the strides were also uniform in the sense that they were ergodic, you still had the error signals distributed non-ergodically. So the error signals were not randomly distributed. The error signal themselves had a history from one to the last stride. Okay. Yes. So when the curve diverges, it means ergodicity breaking. So if the gate cycle is controlled by a computational, prediction, then you will have it. You should have ergodicity, but we found the error signal was actually non-ergodic, which means that the error was not implemented or corrected in the next stride, but the errors were distributed throughout in a cohesive manner with a specific relationship. So this says that error signals are not just random, but the next is we wanted to see what exactly the error signals are distributed. How are exactly they distributed? So we did this fractal analysis, which basically looks at, the scale invariant structure, which I talked about, the, coastline of the Britain, that how the relationship of the deviation is related to the time scale you look for. And what we found was that, again, you can see that even though, again, the more the 2 points, the solid and the, the open circles diverge, the more fractal it is. So you will see that we had only one signal which was fractal, rest of the signals were non-fractal. But when you look at the error signals in the bottom, all the error signals were actually fractally distributed, which means that they had a scale invariant structure. The scale, they were not random, but they were actually following a specific kind of distribution in time, the error signals. Okay. Then we looked at the multifactorial structure. So fractal assumes that there's only one sort of variability. A multifactorial spectrum quantifies the diversity of that variability. And we looked at the width of this. So in this case, alpha, it's a width of the spectrum. So the wider it is, the more variable, the exponent, scaling exponent is. You will see on the top, only the PPS had, wider than surrogate spectrum in the red case. But when you look at the bottom, for instance, and the strides, they both show evidence of cascade-like dynamics in terms of the distribution, which is counter to what we would assume from a computational model, which will be correcting error in the next few strides. So we saw this continuous relationship between signals in a specific fashion, telling that it is not coming out of certain, short range model, but it is distributed across the whole span of the behavior. The interesting thing was, the most interesting aspect, when we looked at the relationship between how good the people were, predicting, the next stride, basically looking at the asynchrony, and the relationship that with standard deviation of, asynchrony or Hurst exponent or the multifactoriality, we found that the more the multifactorial, which means the more the cascade like the process is, the faster people are to predict the strides, which means that this is actually an adaptive distribution. So this distribution is not something which exists, but it is actually related to the performance of people across different conditions.
Madhur, can I just interject? I see that we have about 5 or 6 minutes left. I don't know how far along you are.
I have just one more assumption, which I can hurry. Yeah.
Oh, no, I don't want you to hurry. I thought if you had a lot more, you might want to skip, and I want to leave a little time for general discussion.
Yeah.
Sorry, just trying to help. Thank you.
Okay. We published this in PLOS One last year, which is a very long article because we had to lay the whole framework, and you might like reading it. And we also did something very similar in the context of speech with the reaction time, which we published in Interface before that. The fourth assumption and the revision is that Sensorimotor physiology, it breaks ergodicity, and because of nonlinear interactions, which makes it hard to generalize from group level to individual level. So a lot of work, probably like 30 to 40% of work in my lab is basically simulation based, where we make ground truth models of these physiological signals. In this case, we are generating simple cascades. Cascades I have like tons of varieties of cascades. In this case, what I'm doing is I'm taking a random number, dividing it into 2 randomly, then dividing it again 2 into randomly. And you have this random across generation. So you can think of a neural signaling, a neuron dividing into, branching into different neurons and different neurons and different neurons. And finally, you look at the interactions of all of those. So we actually look at the interactions of all of those in an additive and multiplicative fashion. So first, let's look at the COP. So our center of posture, when we record the ground reaction forces, just look at the C plot. It shows strong ergodicity breaking. This is the real center of posture collected from a human participant, right? It shows strong ergodicity breaking. Then we simulated these time series. On the left is a random time series. On the right, it has a cascade structure. And what we find is that when you produce a cascade structure, it produces the same kind of ergodicity breaking, which your actual signals do. So look at the plot C here, compare it with the plot D here. Okay. So we can break ergodicity in a simulated time series. When you look at the standard deviation, so we divide the time series into different chunks, into different segments. And then we look at how ergodic or non-ergodic is that particular sequence of standard deviation or mean and things like that. What we find is that when you look at the series of standard deviation of the time series, it still breaks ergodicity, which means standard deviation is not reflecting the real dynamics. When you look at the root mean square, even that
Madhur, there's
breaks
a question. Frauke has a big question for you.
Yeah, let me see.
I'm just wondering whether what is simulated and what is human
No,
data,
no.
and if it's
So
human
human
data,
data-
what did the participants do?
So in human data, people were just standing and we record the ground reaction forces.
Mhm.
In simulated data, we produce these cascades numerically. So in
Okay.
MATLAB,
Mhm.
you start with a random number, you generate these cascades with a particular cascade model. And so this is all simulated data. What I'm showing you is simulated data.
Okay.
And in this case, we calculate the standard deviation of that simulated time series over different segments. And you should only focus on the plot D. It breaks ergodicity, the standard deviation, because the two random and original do not converge. When you look at the root mean square, for instance, it also breaks ergodicity. When you look at the fractal of that time series, simulated time series, it does show a little resemblance to the randomized version, which means it restores ergodicity to a certain extent. When you look at the multifractal structure of that, it completely restores the ergodicity, which means that if we measure the multifractality of some time series, of a human time series, physiological time series, we are able to restore that property, which is very important to generalize from group to individuals, right? So here you have a summary. You have the standard deviation, coefficient of variation, RMS on the left-hand side. They all break ergodicity to a certain extent. And you will see the Hurst exponent and TMF actually restores ergodicity. This I'm putting back on the actual COP data. So we showed using simulated data that some measures can restore ergodicity, some measure cannot. And we see the same thing in actual COP data, that these measures can actually restore ergodicity, some measures, and the other measures cannot. So we have published now several papers showing that what exactly are the measures which can identify those properties which will allow your findings to be generalizable from group to individuals. And linear statistics like mean, standard deviation, root mean square, or any analysis which has simple linear variance at its core. For instance, eigenvalue decomposition, singular matrix for like synergies and things like that, they won't be generalizable because they are looking at a linear variance that is not ergodic. But when you're capturing variance which is non-ergodic with the statistics which has non-ergodicity built into that, then you can find generalizable, stuff. So basically that was our fourth assumption. Now I'm not the only one doing all this work. So NASA has adapted tensegrity structures, so a lot more new robots are tensegrity robots because they're omnidirectional. You can put a payload in between. They can stand a lot more forces. So much better. This is a crude assumption, very crude picture, but people are going towards tensegrity prostheses because they work very similar to human body in terms of their compliance. So people are trying to model new prostheses based on tensegrity joint or tensegrity kind of dynamics within that. And we ourselves are supported by NIH right now to identify these networks in the body and use that for rehabilitation, to characterize aging-related disorders and different diseases. So currently in my lab, you have these tasks going on where people stand on force plate on a balance board, which has not been shown here. And they are solving this whole size, trail making task on the screen. So we have a cognitive component. They have to balance the body. And the wobble board is very difficult to balance, not shown in this picture. And we record the ground reaction forces, we record their eye data, we record the laser trajectory, we record every possible dimension. The next experiments will also include FNIRS from the brain and EMG from the muscles. And we are trying to come up with a unifying network models of the whole brain-body coordination, where we talk about the cognitive component, in this case, solving this task, and see how the network interactions across the body influence your ability to do the task faster or slower. So for instance, in older adults, we expect, when you have perturbed stance, your body network will divide into two modules, which will interact differently compared to, simple standing and things like that. And simultaneously, we are working on these methods for others to make available. So for instance, we have been publishing a lot more tutorials recently where where all the codes are available. You can use these analysis for your own work for each of these analysis and, look at all the parameters, what are the assumptions of these analysis and things like that. We have success in identifying Parkinson's from posture, for instance, using this analysis. You can see on the right-hand side, the red one indicates highly non-Gaussian behavior at certain time scales in Parkinson's disease. We are also able to identify healthy people's behavior across different contexts, task constraints using the same analysis. And our latest breakthrough is this paper where we have shown that we can create almost any kind of physiological signal using a combination of additive and multiplicative interactions. We can create any diversity of signals by controlling how much additivity and multiplicativity goes into that, and we can explain simple diseases or breakdown of physiological systems as a change from multiplicative to additive interactions to certain stuff. So we are taking it to the level of biomarkers. We're using the statistics to identify better biomarkers, at least an initial attempt. So we had a little success in heart rate so far. I will skip this. Yeah, so that was all.
We have to wrap up.
Yeah.
All right.
Any questions? Yeah.
Thank you so much. Thank you, Madhur. If people are willing to stick around for a little while longer, any general comments or questions? This is a very different style of work than what we do in our lab, where we tend to be more focused on picking up, simple patterns in more constrained experiments and not modeling to this level of specification. But this is very, very exciting. And Madhur, you are exciting. You're a very exciting new person on the horizon here. I'm very enthusiastic about what you've presented.
I really appreciate it, and I also want to, give a disclaimer that, see, I'm not tied to any theory and any politics or anything. So it's like, this is what is fascinating me, and this is something out for people to look for.
Yeah.
I might be wrong, as every scientist should be willing to accept. But I also am not afraid to, be radical. So
No. And this is for those who don't know. So Madhur is coming out of sort of a tradition well known in psychology from Scott Kelso and Michael Turvey and others, looking at so-called complex systems, dynamical systems, fractals, the study of chaos and things like that. Broadly speaking, that's the tradition that Madhur is working in and pushing forward in some very, very exciting ways. It's so cool to be able to use your general approach, Madhur, to characterize in a way that's somewhat reminiscent of the additive factor method, like looking at sort of how many subsystems may be contributing. It's reminiscent of that. I worked at Bell Laboratories in Saul Sternberg's department, actually. And of course, the additive factor method is long gone, precisely along the lines that you've been focusing on, which has to do with massive interactivity, parallel processing, and so forth.
That's some beautiful work which came out, with that additive, that paradigm at that moment, like beautiful experimentation. I won't deny that.
Yeah, I know you appreciate it. Prof, why don't you comment out loud, please?
I heard the inverted pendulum, and we were trying to apply that to some of our data, but then I was like looking at all the equations and didn't know what to do. So do you have any recommendations on, like, is there already implementation of it so that I don't have to convert?
If you're looking for a biomechanical, engineering-based inverted pendulum model, I mean, it's all over the place. You look at OpenSim, for instance, it is completely based on inverted pendulum dynamics, where we actually simulate, human walking or something like that, or posture. when you're trying to model a time series, for instance, of like posture with the inverted pendulum, along with like different control systems. There's work by Asai et al., the Japanese group, Nakamura et al., published in PLOS One, PLOS Computational Biology, where they have intermittent systems where you have feed forward and feedback processes, and they interact together, and they lead to certain patterns which resemble Parkinson's and humans. They're based on inverted pendulum, but they are simulating the dynamics and not the mechanics. So it depends on what your interest is. Are you interested in musculoskeletal modeling? Are you interested in dynamical modeling? Or you're interested in, even higher order, how it responds to a perturbation? Depending on that, you will have different softwares and different tools.
Okay, so
I'm
it's even
at
more
the level of-
complicated than I thought. Like you wanted to predict like under which conditions people sway with a particular frequency and amplitudes, but given like constraints on like how the joints can move.
So I would say there's work by Nakamura et al., which definitely it has ability to input the mass and the height and some parameters which it simulates really well. It does not simulate the real world effects, but at the same time, it can simulate some effects of more mass versus less mass. Qualitatively, you can look at things. It does really well with that.
I think we should wrap up because we have gone into people's weekend more than we should have. Madhur, this was just great. And Madhur, before I knew more about your work, I suggested that you and I might think about the effects of emotion on movement. And I'm even more sanguine or enthusiastic about that now, having heard about the analytic techniques you have. So the idea, folks, is that the effect of emotion on the way we move is something that's basically never really been studied in a systematic, focused way. And I think the tools you have been working on, you and your colleagues, Madhur, provide a very, very exciting way to probe the effects of different mood states on performance of the very same task with very sophisticated modeling. So I hope you and I will continue to stay in touch.
No, of course. I think it might have a resolution to look in, for instance, prospection when you're thinking about the future or planning, it definitely changes your sway suddenly, right? And which is often not manifested in the mean and standard deviation, but at the same time, nonlinear measures are able to identify those. So there's definitely a scope. I agree with you.
Madhur, thank you so very much, and we really appreciate it. And lots of luck. And thanks everyone for being here. And Ari, I'll see you in 15 minutes.
Thank you so much, everyone. And it was my pleasure.
Thank you again. Much appreciated. Bye-bye.
Hey, David, do you have a quick minute?
Oh, yeah, sure. Go ahead. Let me stop recording.